Alrighty guys, we are going to do our first section of chapter 7 today, and I've split it into two sections. So you're going to be paying attention to the parts in red. That is what will count towards your notebook check. There's a lot of steps in cellular respiration, and I'll be honest with you, I'm not going to have you write all those down. There's a lot of really big words in there, and you're not going to understand it anyways if you're just rereading what you write. So I'd rather you listen and pay attention, and you can always go back through the slides if you have questions or want to relook at those terms. So most foods contain usable energy stored in complex organic compounds, such as proteins, carbohydrates, and fats. This is everything that we talked about back in chapter 3. The breakdown of these organic compounds into simpler molecules releases energy to power cellular respiration. So here is just a refresher. This is what carbohydrates look like. Here are your fats, and here are the proteins. Cellular respiration, by definition, is the complex process in which cells make ATP by breaking down organic compounds. Only autotrophs, such as plants, use photosynthesis to convert light energy from the sun into chemical energy. I don't know why that is still there. Both autotrophs and heterotrophs undergo cellular respiration, so that's really the first big note that you're going to write. This is a biochemical pathway, meaning that the products from one reaction are going to become the reactants in the next reaction. So here we have cellular respiration. It's done by autotrophs and heterotrophs. When you go through cellular respiration, you give off carbon dioxide and water. So every time you breathe out, you are exhaling carbon dioxide and little water molecules, which is why it is so important right now that we wear masks, because you are actually spitting out little bacteria and little viruses every time you talk. Now, those two things are going to be used in photosynthesis, which is only done by plants. They're going to take in some light from the sun, and they're going to give off organic compounds like glucose and oxygen. And we are then going to use that in cellular respiration, and they're going to give off carbon dioxide. Now, the cool thing here, if you notice, there's autotrophs at the top and there's autotrophs at the bottom. So plants could survive without us. They do not need us because they make and use both carbon dioxide and oxygen. We, on the other hand, only use oxygen, and we give off a lot of carbon dioxide. So we could not live without plants because we need their oxygen. Cellular respiration is a little bit confusing, so I'm going to take it kind of slow, and don't worry so much about these really big terms. This is basically what it looks like here. We have glycolysis, and you'll get ATP from that. And then we're going to go into the mitochondria, and mitochondrion is just like the singular form of it. We'll do the electron transport chain where you get the majority of your ATP. Glycolysis occurs in the cytosol or the cytoplasm, which is just that jelly-like substance inside of your cell. Funny little tidbit here, photosynthesis is just cellular respiration said backwards. So many of the reactions in cellular respiration are redox reactions. One reactant is going to be oxidized, meaning that it loses an electron. Now, when you lose an electron, electrons have a negative charge. So if you're getting rid of negative charges, that means you're going to become more positive. And one reactant is going to be reduced, so it's going to gain those electrons that the other one lost. If you're gaining negative charges, that means you are actually becoming more negative. Here's the equation for cellular respiration, and it is photosynthesis backwards here. We're going to have glucose plus oxygen yields carbon dioxide plus water and energy in the form of ATP. If you push this arrow the other way, that's photosynthesis, where you have energy plus water and carbon dioxide is going to yield oxygen and glucose. That's the overall equation for cellular respiration. However, it does not show all the processes that occur. Please, again, don't get intimidated by these big words. I'm going to run through some of the really common abbreviations that you're going to see and hear me say. So G3P is glyceraldehyde 3-phosphate. ATP, you're familiar with that one. It's adenosine triphosphate. ADP is adenosine diphosphate. NAD plus is nicotinamide adenine dinucleotide. And FAD, I don't know, that one always sounds fun to say. It's flavin adenine dinucleotide. So glycolysis is a biochemical pathway, meaning that the products are going to be used in another reaction, in which one 6-carbon compound of glucose is oxidized to produce two 3-carbon molecules of pyruvic acid. Glycolysis occurs in the cytosol of the cell. So here is basically what is going on with glycolysis. You can see there's a few different steps. Energy investment is up here, energy payoff is down here, and this is what you should get. Step 1. 2 phosphate molecules are attached to one molecule of glucose. This forms a 6-carbon compound with a phosphate attached to each side. The phosphate groups are supplied by 2 molecules of ATP, losing a phosphate and forming 2 molecules of ADP. Every time you break a phosphate off of a molecule of ATP, you are releasing energy. So that's the first step here. We have our glucose molecule. We're going to add 2 ATPs. We're going to take the phosphates off and stick them on the ends. In step 2, that 6-carbon molecule we had is not very stable, so it is just going to crack right in half, right down the middle there. So now you have 2 3-carbon molecules, each with a phosphate on the end. The 2 G3P molecules are oxidized, meaning that they're going to become more positive. They're losing electrons, and they're going to receive a phosphate group to the side that is lacking one. The oxidation of G3P takes place at the same time as the reduction of 2 molecules of NAD+, forming NADH. NAD+ is similar to NAD+ in photosynthesis. Both accept electrons during a redox reaction. So if you're getting rid of electrons, somebody has to be there to accept them. So here we have our 2 3-carbon molecules of the phosphate. We're now adding another phosphate to each end, and NAD+ is turning into NADH. The phosphate groups added in steps 1 and 3 are removed, and it's going to produce 2 molecules of pyruvic acid. Each phosphate group is combined with a molecule of ADP to make a molecule of ATP. So that's what's happening down here. Those phosphates are going to be put back on the ADPs, giving you ATP, and here's your pyruvate. 2 to 4 ATPs are going to be produced, which is hardly anything. So you're going to actually produce 4 molecules of ATP, but every time you go through glycolysis, you use 2 molecules, so you only have a net gain of 2 ATPs. So that's 2 that you can use as energy to do whatever you want with. The other 2 go back in so that you can create more energy. We're also given 2 molecules of pyruvic acid, 2 NADHs, 2 molecules of water, and 2 protons. When oxygen is present, cellular respiration continues as pyruvic acid and enters the pathway of aerobic respiration. But when oxygen is absent, the cells can convert pyruvic acid into other compounds in the cytosol, but no more ATP is going to be produced other than those 2 that you got from glycolysis. So fermentation is going to occur when oxygen is absent. And there are 2 pathways that you can go through. It can be lactic acid fermentation or alcoholic fermentation. Now, we can only do lactic acid fermentation. We cannot do alcoholic fermentation. So don't go around thinking, okay, if I try to hold my breath so that oxygen isn't present, I'm going to get intoxicated, because your body isn't going to do that. You are not going to produce ethyl alcohol without oxygen, nor with oxygen. It just doesn't work like that. So here is glucose, and we have our pyruvates, and we're going to be making them into lactate or ethyl alcohol. So we'll talk about lactic acid first, because that's one we can do. It's a type of fermentation where an enzyme converts pyruvic acid into lactic acid. So the NADH is turned into NAD+, the hydrogen atom is lost from NADH, and a free proton is going to be used to create lactic acid. NAD+ is used in glycolysis, where it's made back into NADH. This helps glycolysis to continue in the absence of oxygen. So you're still going to be able to get a little bit of energy, but not a lot. The 2 free electrons from the hydrogen atoms are combined with pyruvic acid to form lactic acid. So for every 2 molecules of pyruvic acid, you're going to get 2 molecules of lactic acid. And basically all it does here is get rid of this double bond and add a hydrogen to it. So you go from glucose to pyruvic acid to lactic acid. Uses of lactic acid, it is kind of important. Milk naturally ferments over time, spoiling, and while that's not necessarily a good thing, it is one of the outcomes of having lactic acid fermentation. And fermentation is also used to produce cheese, buttermilk, yogurt, and sour cream. And lactic acid fermentation also occurs in your muscles during strenuous exercise. This is because oxygen can't get to your muscles fast enough. So you're running really hard or you're really lifting weights and you just can't breathe in fast enough. And that buildup of lactic acid in the muscles causes muscle pain and cramps until that lactic acid is transported to the liver where it's converted back into pyruvic acid. So this is what a completely healthy muscle cell looks like, and you can see it's nice and pink, it's kind of plump, and you can see the individual fibers. That's what this is sticking out. It's a muscle fiber. Whenever you have lactic acid fermentation occurring, it looks like it's degenerating a little bit. It's kind of eaten away. It doesn't look too great there. There's some holes in it, and that's why it hurts so much after you do some really strenuous exercise. It takes a couple days before your body uses all that oxygen that you're breathing in and can replace your muscles and make them look like this again. Alcoholic fermentation is used by plant cells and unicellular organisms like yeast to convert pyruvic acid into ethyl alcohol. So like I said, we can't do alcoholic fermentation, but plants can. The steps are similar to that of lactic acid fermentation. So a molecule of CO2 is removed from pyruvic acid, leaving a 2-carbon compound. Just as in lactic acid fermentation, NADH is converted into NAD+, and a free electron is present. And then we have the 2 hydrogen atoms added to the 2-carbon compound to form ethyl alcohol. It's important because yeast are a type of fungi, which means they cannot produce their own food. They are heterotrophs. When given fruits or grains, yeast releases carbon dioxide and ethyl alcohol, and this allows us to make things such as beer, bread, and wine. And there's a nice little piece of bread there, and you can see those holes in there. That's actually from the carbon dioxide being given off. That's where I'm going to stop for today with our first part here, and we'll pick up next time with part 2.